So, as it has been, I suppose, shown by Professor Smita, is that in fact, when we want to produce some specific metabolite or specific extract, we can develop some artificial way of culture, of plant culture. We can have some in vitro cultures that concern our plants, what we call vitro plants, okay? So here you have all the plants with all the organs that can lead to produce several types of molecules. Or you can shift to more suitable system, the unique cells, cell suspension, callus before and cell suspension after, which can also very easy to manage because you can scale up the biomass in a very good manner by using some bioreactors. Unfortunately, in sometimes, now you know why, has the metabolic pathway of the metabolites you want to produce are complex, but also needs to involve different type cell in the plant. It is difficult to unlock all the genes able to produce this molecule, okay, to lead to the production of what you want to produce. This is why there is a lot of other manner of trying to find a way to cultivate plants, not in vitro system, but perhaps in a system that is not don't use fields, natural way fields. And it is what it is called indoor cultures. And in these indoor cultures, there is two very important types that are mainly used today. And there is a lot of companies that have now completely integrated this type of mode of culture, okay, in their strategy, and in particular here in India, because I saw I have made some research on the website. There is a lot of interest, yes, but also I think the government here pushed people to try to develop this type of mode of culture. So it was interesting to include this idea in this lecture. So you know that we are now quite about 9 billion people. Here you are the most, okay, the country with the most inhabitants, and 1.4, I think it is now the good number at this moment. So you are completely concerned by this idea. Why do you? I have chosen specifically, a photo of Indian peoples, but you see, I could also have put here a photograph of Paris at Christmas time. It will be exactly the same crowdy people. Okay, so there is not, it is not just, it's just a look for you. So here you are main concerned, but it's not only in India, it's over the world. Okay, we are completely concerned by the question of how we can produce enough plant biomass to sustain food, drugs, cosmetic, and also pleasure, because we want to buy flowers every time. Okay, so by this way, the question of the arable areas is of very importance, because in fact we have almost reached the maximum surface, okay, we can use on our planets without endangering what the forests are now. But what it is the problem is that we see that the preservation of the forest is not exactly for each country the first priority. So it could be a problem for the future. So it is important to develop new way of cultivation that is completely independent from these arable areas. And in fact, in the region where this land, and in particular also water, because always there is these two parameters that need to be taken in count, when they are becoming scarce, they have turned their mode of production to intensive high idling farming methods. All it is written in this sentence. High idling means that we have lots of production with a very high yield of production, not only biomass, but also qualitatively, the product. But it is with farming methods, it is in building, not on the soils. And this technology are diverse, and one of them, hydroponics in general, have been already well developed, and in fact they match with all the criteria that we need to complete, means healthy, affordable, sustainable, and also biomass plants. And one of examples, the most interesting one of the example is the example of tomato, where in Europe and also in Asia, Pacific region, it is one of the most sustainable culture through hydroponics by 2028. So means that now it's quite well established, this mode of culture for several vegetables such as tomato. So what are these hydroponics? Little bit history. It's not a so new method, because in fact it comes from Aztec. In fact, at this moment, Aztec had established an original mode of culture. What it is named chinampas, means floating gardens. Okay, that was organized like that. In fact, they have they put some soils. Okay, it was with soils, but a thin layer of soils. Okay, and they were, you see, put in sort of back of wood that have been organized. Okay, and so they are floating, and they by this way, all the plants that are able to grow on this type of soils have their roots that are in contact with the water downstairs. So it was a way, okay, to produce several types of plants, such as, for example, maize or different other plants, herbs, more herbs too. And by this way, they have organized lots of type of floating gardens around palace and small village and so on, in order to be completely independent, to produce what they want as food. So, if you take in account this type of technology, we haven't do this floating garden on our lake, but we can modify completely this vision by making some artificial, system that lead to the same objective, production of plants without any soil. This is what we call hydroponics, that are processes where soils is completely replaced by aqueous solution. Waters, obviously, but also waters in which you have added lots of nutrients that is a way to feed plants with what they need, when they need, and how many they need, to be sure that they are in a better condition to grow and also to produce organs and also molecules in the cells. So, by this way, there is a lot of different processes, and I will describe them after with you, that have been established in order to reproduce this type of ancient traditional way of culture. And what it is now also interesting is that all the climate is also completely controlled, because here you see your garden are outside. So if it is raining, if the time becomes cold or dried, it could be difficult to manage. But we have completely avoid this type of problem by putting all this mode of culture inside, building, inside room, in order to control all the parameters, hygrometry, photoperiods, temperature, all the abiotic, parameters are completely normally controlled in this case. So, and how we are now with a modern system, all these parameters are always controlled, and they are completely linked with several sensors that indicate at each time how the situation of your farm is, and how your plants are obviously in what condition they are growing. So here you can observe that your plants are growing on substrate, sometimes synthetic one, but what it is interesting is that all their root system are completely free, and they can be in contact or in the water. We will see that there is different type of compartment. So what it is interesting is that in this condition there is two major advantages. The first one is that they have a very shorter growth cycle. So means instead of waiting 2 or 3 months to have your final plants, only 1 month will be sufficient to have the same amount of plants. So means that during one season or 1 year, you may have lots of cycles, and so you can harvest plants all during the year. It's in continuous. And because by this way, as all is under control, you are not conditioned by seasons. You are completely free from the season. So it's quite very interesting. But not all the plants can be also cultivated by this way. We'll see with the hand. So what are the different techniques that are used? And techniques not, but systems, it's better, that can be used for this type of culture. The first two one, more simple, are what it is called deep water culture. This deep water culture, as you can see here, it's quite simple, more comparable from the Aztec system of floating garden, where plants, are put on a sort of small layer of film, they are put inside, and their roots are completely immersed, in the nutrient solution, and only a system of aerator diffuse some oxygen in order to be sure that the water is well oxygenated, and the solution, it is directly nutrition solution. This is a more simple system, but after they have been increase the performance of this system by proposing different other experimental systems, such as, for example, the drip system. In this case, you can see that plants are not immersed, roots are not immersed directly in the nutritive solution, but they are put in some of artificial soils. It can be gallery, it can be different type, depends of what you want. Mainly natural one, because it's not necessary this support gives some nutrients and so on, because all will be all the nutrients are in the nutrient solution. So these soils, artificial soils, need to be completely natural. And what you can see here, it is the nutrient solution is completely separate from the plants, and it is a pump that feed all these artificial soils by this nutrient solution. But this is a quite simple system. Another one, and I will describe more film with more detail this one. It is what we call nutrient film techniques, and FT. That it is quite a little bit more complex, because as it is quite similar to this one, but with one difference, is that in fact your plants are also on neutral soils, which is not but you see here there is a thin layer of nutrient aqueous solution that are in contact with part of the roots. And this solution, okay, is not it's not stable. There is a cycle, a circulation of this nutrient solution that is assumed by a specific pump. Okay, and by this way you have a closed cycle, okay, of the water. You don't need to add external water or external nutrient solution. So it is interesting because it is very economic. Okay, after that you have two other, and it is the last one, two other types of system. This one, that it is called ebb and flow. It is quite exactly the same as NFT, but you see here, it's not just a thin layer of nutritional solution that are in contact with the roots, but it is a more important one, and depends in fact of the plants you want to cultivate by this way. Depends of the area of their roots. If they have some small one or if they have some big one, you can imagine that you have to adapt different protocol depending of the architecture of the root system of the plants. And the last one could be also very interesting to detail, and we'll do that after. It is what we call more aquaponics, because here it's very nice. You have to combine not only production of plant biomass, but also you can produce some fish farming, okay? So you can combine production of meat and fish and vegetable in the same times. So some point concerning at least two of these systems. The first one I said to you is the NFT, which is perhaps one of the more simplest and effectiveness system that is a very useful methods. So now there is a lot of companies that have adopted this type of protocols. There is a lot of advantage of this protocol. I like this presentation more perhaps the first one I show you, because you can see here that you have plants that are in this synthetic soils. The roots are here, and they can be also directly in the nutritive solution. You see they are not completely horizontal; they are with a small declination. This could be a way to naturally, okay, permit the cycle of the nutrient solution, and only a pump is necessary, okay, to re-inject some nutrient solution in in this upper, okay, compartment of the systems. And this system can be some very long tubes. It's not necessary to be big, okay, bag. It can be small closed tube, and you can have a very, very, very large amount of plants on this tube. So I will show you some photo. You may have some farming that are very, very, very big only by this system. So what are the advantage of this particular techniques? Here they are listed. The first one, I have already said that to you, is that the use of water and nutrients are completely efficient, because you are not obliged to integrate new nutrient and new aqua solution during the culture of the plant. If you have well established the composition of your nutritive solution at the beginning, it will be enough for all of the culture. Depend of the plant, depend the time of the plant need to grow. Even if it is faster, you need to adjust completely this nutritive solution to the type of plant, okay, you have to cultivate it. It's like when you are in vitro culture, you have to adapt the culture medium to the type of plant you want to stimulate at the growth system. So here it's the same. So it is not always exactly the same nutrient solution used for every type of plants. So it is known that this system, it is one of the most water efficient hydroponic system available. You know now why. What it was already said, faster plant growth and higher yields. In fact, we can divide at least by 2 times of the growth cells, perhaps more, I don't know. No? 2? It could be, yes. Okay. So, but it is lots of time waned for people that are producing plants. Okay. The third one is what we call space-saving design, because this system is really compact, so you can produce a large amount of plants in a small surface, because you are not. It is not necessary that all of these systems are at an horizontal way, but you can have, okay, different system on a vertical way. So you may construct a building, okay, with this type of system of culture. So it is quite very interesting to organize, and so you can maximize space, and you can make them ideal for what we call indoor farming. This is very interesting. Also this, always there is a question. Reduce risk of soil-borne disease. It is obvious in the nature there is a lot of phytopathogen in the soil and so on. So you are obliged to be careful to your plants, but also to apply what we call pesticide, insecticide, different chemicals. In fact, that it is complex to manage, because it can be dangerous for us too, and not only for the pathogenesis of the plants. Okay, so in this case, you see, you can completely minimize also the use of what it is named a pesticide, and it's not sterilized condition as it was with in vitro culture, but as it is under control, you can have more. It is quite easy to verify that there is no, for example, fungi or so different other type of pathogens in this system. So you can reduce completely pesticide and others. And what it is also the case, I said, you can control completely the growing environment. So means that you adjust exactly time, intensity of the light, of the temperature and hygrometry needed for each plant to optimize, okay, the growth in these systems. Okay, there is some advantage, but there is also some disadvantage. Not all is perfect. No, not all is perfect. So what are the disadvantages of this technology? Firstly, you need to be careful, so you have to take a look every time, okay, because in case of, don't know why, pumps stop to function, all the system will be completely disorganized. And in particular, if there is no pump, then we have no water, and if you are not concerning by this at that moment, root can dry, and after you can lose all your culture. So you need to be careful and to control, okay, every time, and your system. So you have now there is some electronic control that alert you on your phone. There is something wrong on your pump, and you can go and you know exactly the number of your pump, and you can go to see what happens. But means that you need to have this type also of monitoring in order to control the good functioning of mainly the pump, because it is the only mechanical part of the system. Second thing is if you have not good positioning of your plants, or if you have not adapted the quantity of nutritive solution in this particular compartment, your root not completely emerge as it is necessary to have, so they can dry even if all is making well. So you need to adapt exactly this flux of nutrient solution cycle in order to be always in the optimal, condition of emerging your roots. What I have said is that, in fact, it is a good thing if you have plants that have not a particular big, root system, because it will be not possible to adapt. So until now, the main plants that are cultivated by this type of system are herbs or vegetables of small root systems. But there is now new systems that have been appear on the market that permit to increase the number of plants that could be also cultivated by this methodology. And the nutrients imbalance, you need to maintain this balance of nutrients in a correct manner during all the culture, which is quite challenging because, in fact, at what moment people say you put all and you wait, and 1 month after you harvest your plant and it is finished. In fact, it is not quite exactly the same, because what happens, the plants, take all the nutrients of the solution, so the concentration, the final concentration of the nutrients decrease during the time. So at what moment it can become a negative factor instead of being a positive one. So you have to control every several days, and particularly at the first steps of the culture, you have to evaluate the quantity of nutrient that remains in your solution. In case of necessity, you can add some new nutrient in the solution in order to maintain at least, good concentration. It is quite the same for in vitro culture. You have to subculture them, each, I don't know, the frequency depends of the plants, in order to put your in vitro plant or in vitro cell always in solution with enough nutrient elements to be sure that all the culture will grow correctly. So this is important, because in case of no, you may have some surprise concerning particularly yields. Growth, yes, but yield at least. The second system I want perhaps to give you some details, it is the aquaponics systems. It's very funny to see, because you have a very nice system depends off. And is what I say to you is that, in fact, you use the same system as we have seen before, means you have your plants in a specific box, which is where the roots are quite immersed, in a nutritive solution. But this nutritive solution is quite particular obtained, because this nutritive solution come from specific fish culture system, fish growing system, because what happens is that, in fact, fish are covered, by several microbiotes, including several bacteria that are involved in nitrification. And in fact, this bacteria can act as plants' organic nutrient source. But they can modify the waste of fish, to create continuously some nutrients for plants. So, natural way to produce the nutrient. And then the solution in which fish are growing or multiplying also, is used by a pump to feed, the plants cultured as hydroponics systems. And after, it's like a cycle too. This nutrient solution come back to another type of compartment that is linked with the fish compartment because this plant releases lots of nutrients that are interesting for fish. So it's quite a virtuous cycle between plants and fish. One feed the others, and it is always the same. And by this way, it can be completely autonomous. You can see here, instead of having a pump, okay, that permits the circulation of the nutrient solution for the fish or for the plants, you can be completely autonomous, okay, for both of animals or plants nutrients. Some of specific fish are well adapted to this system, in particular tilapia, trout, or catfish. Okay, so if you go to see on internet different type of this organization, you can see these three type of fish. And the only things you have to do is to have also different probes here to maintain water pH, hardness, alkalinity, okay, in a constant manner in order to maintain the environment of a fish, for the fish in a good way. I don't know if you have an aquarium at home. If you have one, you are obliged every week or every 2 weeks to control, okay, different parameters in order to be sure that your fish are in a well environment. It is exactly the same here. It's a sort of a large and big aquarium. Okay, so this is the only things you have to see, and also you have to take care about the growth of your plants, and when they are arrived at the maximum of growth, okay, to change all the system. So Hall also is under different monitor, in particular monitor oxygen level, because for fish as for plants, it's very important to prevent stresses, obviously, and also suffocation of fish. It will be a bad thing. Okay, so there is also some advantage of the system. It is because it is a sort of symbiosis, yes, we can call that, okay, between fish and plants. And it is an hybrid system between aquariophilia and hydroponics, as we have seen before, and it is interesting to have fused both of the two systems together. It is a real sustainable resource management. Unlike traditional farming, okay, which often depletes soils and water resource, okay, as we have found here, water was used efficiently, not only for plants, but also for animals production. Year-round production, because aquaponics allow for years-round cultivation of crops, means that you can make lots of harvest during the year, okay? And it is completely independent from outside, okay, condition, because it is completely under control, under your own control. And this is a way to making a lucrative business opportunity, and some of lots of society have taken this opportunity, okay. There is also a higher crop field. Indeed, this water is very rich with nutrients that are not perhaps completely the same during the time. Depends of the own cycle of the fish. Depend also of the activity of their external microbiota. So means that in fact the nutrient can be also variable along the time, and this could be also very interesting for the plants, because it can also, and I will see that, I will see you that, you can stimulate metabolic production by this way too. You can combine different things to stimulate this. And reduce chemical usage, obviously, you have seen here, you don't need any other system. It is, you don't need pesticide also, and you are more sustainable in a sustainable system. Here, some example, okay, you see, here it is what I have called hydroponics, okay, you see some tubes that are, here they are horizontal and they are, okay, pulled under the others, okay, so you have lots of scale, so it can become very important surface. You can make lots of production during a small time. Here it's the case of Eden Green, okay, which is called an hydroponic farming, vertical farming. And here you can observe that, in fact, if you look at the surface used by this type of hydroponic farming, they have a smaller surface than the equivalent field that will be needed to produce exactly the same amount of plants. And here an example of a big aquaponic organization. Here are the swimming pool with the fish, okay, and here are all the plants that are completely fused with this system to be feeding by the waste of the fish. It is this particular company, Aquahydro. Yes, it's a quite new one, because I saw that before it was a smaller one, but now it's becoming a very important one. And this is interesting to see. And the question is now, this system, aquaponics in particular, could be the future nutrients in space. You see that there is a lot of space program that aim to send several people for long time, several years in the space. So they need to find a way to produce foods, animals, and plants in order to have enough foods during the time. And it seems that this system could be one of the possibility, because as I show you, it's quite completely autonomous, and only need at the beginning to implement first plant and first fish in the system in order to have enough. So they want to take part of this, to take advantage, of this virtuous cycle to apply this type of methodology, for example, for this system. But not only on space, it was just to give a super dimensional system, but also it can be also think for here, depend of people where they are in some space where it is very hard to live, for example, near deserts, near different other extremely different environment. That could be also a way to assure foods, correct food and good quality of food for colonies of humans, in different parts of the world. So here are some examples of currently vegetables produced by this type of techniques. So you have root vegetable, leafy vegetable, bud vegetable. You can have here lots of name. What it is interesting, so tuber, stem, inflorescence vegetable, like cauliflower, for example, and also fruits and pulse vegetable. Here are the type of cultivation that are already adapted to this type of specific vegetables, and are the name here. What it is interesting when you see some of them are considered aromatic plants, but aromatic plants are medicinal plants. Lots of aromatic share with medicinal plants the ability to produce specific metabolites that could be used for application. So it is why now we can observe that this type of methodology of production can be adapted also to produce specific metabolites for pharmaceutical or for cosmeticals or other type of application. Because now we have see the advantage of having this type of mode of culture. This is what I wanted to present you, and it is what we call aeroponics. Aeroponics, it is a culture that mix hydroponics, and in fact to be not in a completely immersed solution, at the level of the roots. Here you can see the classical system of aeroponics. You see the culture are not completely in contact with the nutrient solution, and the roots are not in the soil. They are in the air, okay, in a closed box where the humidity, yes, obviously is quite high, okay, it's not so dry, and with a frequency that have been established at one moment, the nutrient solution is completely spread, okay, on the roots. So you may have your roots that can have a big, okay, amount of roots. Here there is no limitation, and it was with hydroponics or aquaponics systems. So you may have more roots, and also you can maintain them in a dark chamber, means that roots are quite when they are in the soil without directly light, okay, light. So it is a good, interesting, because by this way you can avoid completely development of algae, which is perhaps one of the problems of the both two other systems. You have your solution here that is sprayed onto the roots at regular intervals to be sure that they have always nutrients. And what it is interesting is that finally this system exposed continuously the roots to different parameters such as oxygen, and they can have, in this case, the maximal absorption of nutritive solutions that are sprayed. Moreover, in the solution, you can add some what we can call adjuvants too, that can also facilitate, okay, the infiltration of nutrients directly in the roots when dropped, okay, or sprayed on the roots. And in this case, we can observe that we can have an exceptional growth of the root mass. I will show you some picture. Is that instead you have a normal, normal biomass of roots, you can multiplicate, this by 10 or more than 10, okay, this biomass. And we have seen together that some metabolic pathway occur specifically in roots and lead specifically to specialize the metabolite of interest. So it was also interesting to imagine, to adapt this type of method of production, okay, not for the plant by itself, but for the ability of the plants to produce specific metabolites, and in particular in roots. Okay, so this is very interesting because this methodology have been applied in different process. I will show you, and now it represents a very important market. Okay, so there is a lot of possibility with this type. It was estimated at about 2.7 billion last year, and they are expected to reach more than 18 billion in 10 years. So it means there is a lot of possibility with this type of mode of culture, and in particular into for metabolite production. Just a detail, there is two types of design. It's just a question of techniques here. Depending on the pressure with what you sprayed, okay, the nutrients, nutrient solution on the roots. So you have the high pressure and you have the low pressure system. If the first one is efficient for nutrient delivery, the second one is less. But when you put some high pressure, you may have some problem with roots if they are sensitive to this. So you need to find here also a balance between technical parameters and also sensitivity of your plants. So you can adapt. But it is true that with low pressure system, you may have a more controlled way to apply different molecules or nutrients that are in your solution. So here, I want to explain you the example of one company, a French one named Plant Advanced Technology, that have been created now 10 years ago. It was two colleagues of me that have been at the initiative of this company creation. So I have followed lots of things that I have done in this company, and they have adapted completely this process of aeroponics, okay, to produce a specific metabolite that can be used for pharmaceutical or cosmetical application. In particular, the two researchers that have been created this spin-off, okay, of their lab were involved in deciphering metabolic pathway of coumarin and furocoumarin, for example. But they were also, as I am, and as Professor Smita is specialist in plant cell biotechnology, hairy root biotechnology, and different technology on the side. They have, okay, combined all of this knowledge to propose this type of technology to produce this specific metabolite specialism. And now they have some patents, okay, to protect their discovery, and in particular one that it is called the PAT exudactive, and I will show you what happens. So here it's one of the picture I want to show you. This is the root biomass, okay, produced by this way, okay, for different plants. So the principle is quite easy to understand. You have a plant, you have found a plant that is able to produce a very interesting metabolite, specialized metabolite. You know that this bioactive molecule can be produced in bigger amount because you have, for example, produced plants or selected plants that are able to produce. First problem, if my biomolecule is produced in leaf, I need to find a way to permit, okay, the migration, okay, of this molecule to the roots. This is important because at one moment, or they need to be synthesized in the roots, or they need to be transferred from the leaf to the roots easily. Okay. And after that, what it is interesting is that by this method, it is not an invasive one because it's not necessary to cut the roots in order to collect or to harvest the molecules. In fact, the idea is how to perform exudation, okay, of this molecule by the roots, because naturally roots are able to exudate lots of components in order to have some interaction with their environment in their soil. So they are naturally able to exclude, okay, different metabolites. So we take advantage of this natural comportment of the roots in order to say, okay, the biomolecule are produced leaf or roots, but after, instead of being accumulated in the roots, which needed to harvest the roots in order to extract them, the idea is how to stimulate the exudation of this molecule in order to collect the exuda and to maintain for a long time in this process all the roots to produce in continuously, okay, the molecules. This was the start point of the story to the creation of this particular company. So it was exactly what they have done. They have developed. Here is the workflow they use. So at the beginning, they have to select the plants, okay. As it is completely in a controlled area, it is not outside, okay. You have seen it's in a controlled area. Sometimes it is possible to use some transgenic plant in case of we need to have modified a little bit some metabolic pathway or different things. So, but you have to select the good plants. You have after to make some aeroponic culture of plants, traditional one without anything. And when you have enough, okay, biomass, root biomass, you can enter this plant in this workflow in order to produce the metabolic pathway. So the second step is to stimulate, okay, because naturally they produce some, but sometimes it's not enough. So we need to elicitate, okay, the plants, as it is also the case in vitro system. So by this way, these plants, okay, it's like it's an automatic system, okay, are stimulated by a spray of elicitor, okay, directly on the root. This spray induce or elicit this metabolic specialized biosynthesis in order to increase the concentration of this metabolite specifically in this in the roots. And after that, we let them for several hours or days, but not so many times. It's not necessary because all the system of the pathway is already present. It is only a way of stimulate them, okay? And after that, when they have produced the molecule, they are bathed, okay, in a aqueous solution, and all the little drops that are on the little exudate drops on the roots, okay, are mixed with aqua solution, and you can collect, okay, all the molecule without necessity to harvest, in fact, the root biomass. And by this way, plants can be re-injected in the workflow, means that as they have enough roots, they can be stimulated a second time, and they can produce a second time, a third time, a fourth time. So you can use them for a long period, not for many years, because at what moment it became difficult to maintain. So you have to renew, okay, the raw material of plants, but it can be very interesting at the end. And what happens with this solution that are now with the biomolecules needed, but they are collected and they are purified, depend of the application, and depend of the molecule. They can be formulated to become a drugs or to become a cosmetical systems. So this is a workflow. It's very efficient. For the moment, PATs have at least, I think, 2 or 3 molecules that are produced by this way at an industrial scale. And there is on the market. They have done last year. They have also proposed a new one. So here are the advantages of the systems. But I think if I ask you the question, you find them very easily. Okay. So this is a sort of unique technology on the market. They have patented it. Okay, so, but there is a possibility to develop several similar technology. It's a renewable treatment. You have seen that we can use plants not only one time. Non-destructive. Preservation of the molecule of interest. Yes, because you collect them just after exudation and you can manage them directly in the process, a downstream process leading to purification of the molecule. Yes, you can have a very important yield, in particular by increasing the surface area between root and the final solution. It's very nice photo here. You can see, okay, all the drop of the exudate in which there is lots of metabolites, okay. Necessary for a large quantity molecule produced and in a very short time, because the process of workflow is not so long. Some days it is sufficient. The longer time is to have enough biomass, so the culture of the plant at the beginning. You can explore the natural wealth of the roots, but you can also, as I said, modify different plants in order to have specifically production of molecule in roots. The secret, I can't say, and you can't know, is how to stimulate, elicitate, and to facilitate exudation it is a patent they have applied for a part, and I think a part is already a secret. But you see, it can be done. And by this way, what it is interesting is that you can have access of high pharmaceutical value, because the disadvantage is that this system is quite expensive. Okay, you can imagine, but not perhaps more expensive than a cell culture in bioreactor. Okay, it's just another type of technology. So it means that it is interesting to develop this type of methodology if you target specific very high value molecule, and in particular it is for pharmaceutical markets. But we see now that there is a lot of big company of cosmeticals that start to be very interested not only by extract but by specific molecules, in particular molecules that can replace some preservatives, okay, that are known now to be perhaps dangerous for our health. So they are very attentive to this type of methodology, including cell culture, root culture, and different other methodology I will show you next weeks. And in fact, the cost became not so a limitation of development of this type of process. Depend of the molecule we want to obtain. And yes, it was last January, okay, in 2024. This company present one of the last ingredients now in the market, okay, that have been now proposed to different companies. Okay. I have detailed this example because I think it is one of the most interesting, showing that how combining plant technology culture, knowledge of metabolic pathway, elicitation, extraction can be fused in one process in order to use quite a natural system, okay, to have enough amount of specific molecules. And what it is also interesting in this case is that with an entire plants, and with in particular these roots where you have all the type cells, epidermis, parenchyma, and so on, you have all the metabolic pathway established in different parts of the cell. You can produce very complex metabolites, specialized metabolites, that it is quite difficult sometimes to obtain directly in one cell, one unique cell. And this is why it was interesting to see that people that are expert in biotechnology have combined, okay, this type of technology with the knowledge of production of this complex metabolic, specialized metabolites. So here, sorry, it just, I think it is my last one, just to compare, for example, between aquaponics and hydroponics, which if you have now the choice to develop a new company based on this type of culture to produce several plants or several metabolites, because it can be also, you have seen, an objective not only for food but also for pharmaceuticals, for example. You have here different type of criterion that have been compared. For example, aquaponics is cost effective, while hydroponics is less cost. This is normal. You have not exactly the same involvement of different systems with fish, with plants, and so on. The productivity is also comparable, means that if in aquaponics you have a higher production than in hydroponics, so you have to find the balance between both technology you want to develop in this case, and also all what is required to maintain in a good state all the process. Okay, so it is only requires a little maintenance in this case, perhaps more maintenance in this one. Okay, So depends of what you have possibility to invest at the beginning. Depend of the surface. Depend of the objective you want. You can have now different type of culture method, okay, to produce plants for several applications. And here, it's not me. I have taken this on one of your journal, Times of Agriculture. Okay, it's an Indian one. They have spoke about some of companies, Indian companies that have taken advantage of this technology, okay, to develop a production of several plants, mainly for food at this moment. But as I show you, it could be also extended to several other applications, including production of several metabolites. So I don't know if you know one of these company.
Yes.
Yes.
Polyphony crops in Tamra.
Ah, okay. So you have an idea of what type of
It's actually a drip system.
Okay, and what type of plants do they produce? Spinach. Okay, so for food, mainly for food. Do you know if one of these company, because I didn't find information of that, if someone are producing some plants for metabolite extraction after?
No.
Not yet. Not in this case. Okay. So I have finished. Yes, today.